So hopefully you took a minute to read over this question and sort of test yourself and think about the answers. We have a picture here of the stomata being open and the stomata being closed. You can see those guard cells around the edge of the stomata control that. And now, of course, when you close the stomata, that does limit the amount of water loss. However, it will change the composition of gases inside the leaf. And so this question here, when we really think about it, is really more of a review of photosynthesis than anything else, right? So how are the gases going to change on a sunny day when photosynthesis is happening and the stomata are closed? Well, the best answer here is A, right? Levels of CO2 inside the leaf are going to go down, and that's because that CO2 is being taken out of that airspace and being fixed into sugar molecules. Meanwhile, photosynthesis produces oxygen, so oxygen levels are going to increase inside there. And so that's how the composition of the gases are going to change if the stomata are closed. And it's actually that change right there, that shift from CO2 levels to oxygen levels, that create the second challenge of photosynthesis, which is something called photorespiration. Now, the word respiration in there should be familiar to you. You know, that's our form of metabolism where we're taking in oxygen, we are giving off CO2, and you're going to see some of the same things happening here in photorespiration. However, even though some of the inputs and outputs are the same as our respiration, the biochemical pathways are very different. So I'm going to go back to this slide that we looked at earlier. Now remember, we already have here this diagram of the Calvin cycle. So photorespiration is a different pathway, and it starts though with this same starting point, the ribulose bisphosphate. And what happens is that instead of a CO2 being added to that ribulose bisphosphate, oxygen is added. So O2 is going to get added in here. And now what's really important to know about this is that the enzyme that catalyzes this first step in this other reaction here is the same one working over here. Remember, we labeled this before. There's Ribisco, this important enzyme that does the fixation of carbon. It's that same enzyme here that catalyzes this first step of what's going to be the photorespiration reaction. So you are used to thinking about enzymes, and you're thinking about their active sites, and you probably think of enzymes as being very, very specific to their substrates. However, Ribisco is one case where there is a certain amount of flexibility or a lack of specificity of those substrates. So the active site of Ribisco, it can grab a ribulose bisphosphate, and then if there's a lot of carbon dioxide around, it's going to grab a CO2 and add that to the ribulose bisphosphate. But if there's a lot of oxygen around, it can grab that and add it also to the ribulose bisphosphate. So let's keep following this other pathway around here. If we were to incorporate an oxygen, we're going to end up now with a 5-carbon molecule. The name isn't that important, but right, we're just adding an oxygen to it. And then the plant still has this problem of it has to regenerate the ribulose bisphosphate so that the Calvin cycle can continue. And this, I'll show you in a second, but this is a long, complicated pathway that involves many steps to take this C5 molecule and turn it back into ribulose bisphosphate. And one of the things that happens as it's doing that is that CO2 is actually given off and lost to the environment. So this is CO2 that the plant has already worked hard to take out of the environment and attach to a sugar molecule, and now it's being released back out into the environment. So that's why photorespiration, and that's what this pathway is over here. This is photorespiration. Is sometimes thought of as sort of a wasteful pathway. There's another important thing that happens as part of recapturing the ribulose bisphosphate once photorespiration starts, and that is that the cell has to use energy in the form of ATP. So here's ATP plus water going to ADP plus an inorganic phosphate, right? So we're also using stored energy in the form of ATP as well. So there's a net loss of CO2 and a net loss of ATP in this photorespiration pathway. So I'm going to go back now to this, and let's say a little bit more about photorespiration. This occurs when Rubisco uses O2 as a substrate. And it results in a loss of fixed carbon as CO2. Right? So when you think about it, here's the respiration part, right? We're taking in oxygen in this reaction and we're releasing CO2. So it's a little bit like our respiration that way. And now the important thing is this is favored. Obviously, the more, the Rubisco is not selective, right? Whatever gas is more abundant is the one it's going to use. So photorespiration here is favored when there's high levels of oxygen in that airspace in the leaf and low levels of CO2. Right? So when there's high oxygen and low CO2 in that airspace, photorespiration is more likely to happen. And now going back to this, that's exactly the conditions we would create when we close the stomata, right? So if you close the stomata, you limit water loss. However, you're going to create the conditions inside the airspace in the leaf where this kind of, sometimes characterized as wasteful pathway, photorespiration, is going to be more likely to occur. Just for fun, I told you that this was a simplified version of this. Here we can look at a more complicated version of all of the chemical reactions that are part of photorespiration. Here is ribulose bisphosphate, and this is the arrow here that represents the step catalyzed by Rubisco, and you can see here we can either add a CO2 or an oxygen. And when we add an oxygen, it starts this whole big long pathway here that we have to go back through to eventually regenerate this ribulose bisphosphate. And it involves up here the loss of CO2, and there's steps where we're using energy that the plant has stored up elsewhere. So this is a big, complicated pathway. You really just need to know that first step that's catalyzed by Rubisco. Now, often students wonder, what is the basis of this lack of specificity for Rubisco? It seems like this problem could be solved if the Rubisco enzyme just used CO2 as a substrate and didn't use oxygen. Well, we have to actually think back to think through the evolution of photosynthesis and as part of the explanation of this. So here's today, right? Now we're going back in time. So here's 4 billion years ago. And in the early Earth atmosphere, looking at the percent of gases in the atmosphere, CO2, well, let's start with oxygen. Oxygen levels were very, very low in the early Earth atmosphere. So this line here represents oxygen. And then there was this point, about 3.5 billion years ago where photosynthesis evolved. So organisms that could do photosynthesis evolve here at this point. And this is also when Rubisco, the enzyme that's so important for carbon fixation, also evolved. Now, what happens once photosynthesis starts? Oxygen levels start to increase. And so, at that point, oxygen levels increased and increased. They got to a kind of a high point, and then they came down to what we have today, which is about 20% of the gas in our atmosphere is oxygen. Now we can also think of the profile of CO2. In our early Earth atmosphere, CO2 was very high. So here's CO2 as this dotted line. Now again, when photosynthesis evolves, what's going to happen? That CO2 is going to be taken out of the atmosphere and it's going to be fixed and it's going to become part of these organic molecules in plants. And so CO2 levels drop down pretty low. Now, of course, today what we're experiencing, because we are so good at digging up this ancient carbon and burning it and putting it back in our environment, we're seeing this increase in CO2. Interestingly, we think a lot about CO2 in our atmosphere. It's really just 0.05%. But of course, that little bit of CO2 can have a major impact on the climate, and we're seeing the effects of that all over the place. So here's the important thing. When Rubisco evolved, oxygen levels were very low, right? So we're talking about in this little area right here. Oxygen levels were very low, CO2 levels were very high, and so there probably wasn't much of a selective pressure for Rubisco to evolve this selectivity for CO2. Now here, when we ask plants to do photosynthesis today in these conditions, there's a lot more oxygen around, and that creates this issue with photorespiration. Oh yeah, I forgot this was in here. You're going to learn a little bit about respiration and gas exchange in animals also, and you might know that there was a period of time where there were giant insects, around. We don't have them today, but if you want to think about where this insect lived, it sort of goes in around here, right at this time when oxygen levels were peaking. That's the sort of the age of the giant insects, and that's all tied into their form of respiration and gas exchange. So, the two challenges of photosynthesis. One is water loss, right? If the sun is shining, you're going to get transpiration off of those mesophyll cells, and that water is going to get lost to the environment. The second problem is the loss of fixed carbon via photorespiration. So what's a plant to do, right? You can leave the stomata open. That decreases the photorespiration problem, but you're going to lose a lot of water. You can close the stomata, and that limits the water loss, but then you're going to have this kind of somewhat wasteful pathway called photorespiration happening at a higher rate. So what's a plant to do?